Hello, everyone, and welcome to today's live broadcast towards a universal influenza virus vaccine. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars, contributing to the advancements of science through the content sharing capabilities. LabRoots is a powerful advocate in implying global networks and communities. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the top, at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing educational credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of attaining your credits. I would now like to introduce today's guest speaker, Dr. Peter Palese. Peter Palese is Professor of Microbiology and Chair Department of Microbiology at the Icahn School of Medicine at Mount Sinai, New York. His research is in the area of RNA-containing viruses with special emphasis in influenza viruses. Specifically, he established the first genetic maps for influenza A, B, and C viruses, identified the function of several viral genes, and defined the mechanism of neuraminidase inhibitors. He developed the field of reverse genetics for negative strand RNA viruses, which allows the introduction of site-specific mutations into the genomes of these viruses. This technique is critical for the study of the structural function relations of viral genes for ingestive viral pathology and for development of manufacturing of novel vaccines. At present, Palese Group works with Adolfo Garcia-Sarte and Florin Kramer on the development of a universal influenza virus vaccine. Palese was a recipient of the Robert Koch Prize in 2006, a recipient of the European Virology Award in 2010, and recipient of the 2012 Stanford Institute Pasteur Award, and awardee of the 2015 Berking Virology Prize of the Royal Netherlands Academy of Arts and Sciences. Palese is a member of the German National Academy of Sciences, a corresponding member of the Austrian Academy of Sciences, and he is a member of the National Academy of Sciences, a member of the National Academy of Medicine, and the fellow American Academy of Arts and Sciences. I will now turn it over to Dr. Palese for his presentation.
This is Peter Palese. I think we have some problems. I don't get the slides up.
Yeah.
Just 1 second. Okay, here we go. We will talk today about pandemic influenza viruses in the past and in the future, and what it means to really make and try to get a universal influenza virus vaccine. When we talk about influenza, and particularly pandemic influenza, meaning an epidemic which has global dimensions, then we always have to think of 1918. And what this slide shows us is the life expectancy in the United States from about 1900 to 2001, both female and male. And we see that in 1900, the average life expectancy was barely over 45 years, 47 years. And then we see, fortunately, an increase in life expectancy all the way up to 2001. And if we are thinking about a baby girl born in 2016, we are talking about a life expectancy of way over 80 years. The curve, which we see here, however, has one very remarkable dip of about 11 years, and that was the result of the pandemic of 1918-1919 of influenza. So this is an event which has historic dimensions. The life expectancy was reduced by about 11 years and was a result of this influenza outbreak from about November of 1918 to February of 1919. In the United States, we had about 750,000 deaths, and worldwide, we are thinking that maybe up to about 100 million people died during this pandemic of 1918-1919. It's also sometimes referred to this influenza pandemic as the Spanish flu, and this had to do that Spain, so this was during World War I, and Spain was at the time neutral, and the only newspapers or the press could only report from these neutral countries, and Spain was the only country at the time which reported about the influenza outbreak, and that's why it was called Spanish flu, but that didn't mean there was no flu in France, or there was no flu in the UK, just in contrary, because these were countries which were involved in World War I, and there was a lot of influenza. In fact, in terms of the US involvement, more people died in the military of influenza as compared to direct military action. So this is really something which is quite dramatic. And when we talk about global epidemics, pandemics of influenza, we sometimes really think about a virus which was as bad as the 1918-1919 pandemic. The next slide shows us an electron micrograph, and this was taken by a student of mine, Yi-Ying Chu. And what we can see here is a virus which has RNA, it's a segmented genome, and we can see one RNA in the center, and then around this one RNA segment there are 7 other RNAs which are making up the genome of the influenza virus. And then on the outside around this RNA is a lipid membrane which is provided by the host cell, and then there are, very importantly, in this lipid membrane we have inserted the surface spikes which are referred to as the hemagglutinin, and we will see there are different subtypes in terms of the H1, but as I sort of intimated already, we have about 18 different hemagglutinin subtypes, and then we have also the neuraminidase, and I show here one of the subtypes of the neuraminidase is the N1. So this is actually the virus. It is very, very small. If you have a computer in front of you, then it's maybe 20 inches, 15 inches in diameter. And a cell which gets infected by such an influenza virus would be the size of the World Trade Center, the old World Trade Center, or the Empire State Building in New York City. And one of these virus particles can infect the Empire State Building, and within about 8 hours, about 100,000 new virus particles are being made, and you can see that such a virus replication, such a burst of virus replication can actually result in the death of the infected cell. So one of the take-home messages of what our discussion today is that there is a lot of variation. There is a lot of genetic variation, of antigenic variation, and in terms of influenza viruses, and the important antigenic variation is on the outside. So again, we are talking about the hemagglutinin, and we are talking about the neuraminidase, which are the surface proteins on the virus, and that is what our immune system is actually recognizing. And again, a very important point, we are talking about extraordinary variation, genetic variation. And that's actually is the reason why we don't have any good vaccine, because there's a lot of this variation going on. And as you probably know, there are other viruses which are similar in terms of this genetic or antigenic variation. So this is a dendrogram, so the length of these bars here, or these branches, will reflect the diversity of the different viruses. And on the left, we have here the influenza virus diversity of the hemagglutinin, and this is shown here. And very importantly, you always have to look at the bar, which is telling us what the degree of variability or variation is, and in this case it is 5%, so the length of this bar reflects a difference in 5%, and if we look now at the other diversity in terms of viral proteins, we have here HIV, and again you can see here the diversity is quite dramatic. If you compare it to what we just discussed before in terms of influenza, hemagglutinin or HA, HIV is actually a little bit less, but clearly that is also one of the reasons why we don't have yet a vaccine against HIV, because the virus is constantly changing, and even within one patient there can be dramatic differences in terms of sort of the cloud of different HIV viruses. And then we also have on here the hepatitis C virus, the E protein, the diversity, and again you can see here, if you just look at the bar, again this is 5% difference, and one can see here that the different variants of hepatitis C virus in terms of the E protein is also quite dramatic. So this is something we should be very much aware of, that and we will see, for example, we have wonderful vaccine against measles virus or measles, and that has to do that the virus doesn't change, and I will briefly discuss why that is so as we go on. Now, if we look at the diversity of the influenza virus hemagglutinin in more detail, we are just looking here, we have now a different bar here, this shows 7% diversity, and in blue we have the group 1 hemagglutinins, and they are actually now 18 different ones, and then we have group 2 hemagglutinins, and there are 6 of those. In terms of human disease, we have only seen so far over the last 100 years, we have seen H1, we have seen humans since 1918 H2, and we also have seen H3. So these are the only subtypes, H1, H2, and H3, which we as humans have seen over the last 100 years. Now, the epidemiology of influenza viruses is quite interesting. It's complex. So the next slide shows actually the different influenza viruses which are circulating in the human population. And as I mentioned before, we have the 1918, and that was an H1N1 virus, and I will briefly discuss a little bit more what we know about the H1N1 virus. And these viruses were present from 1918 until 1957, and then we got actually a new pandemic strain, which is referred to as H2N2 in 1957, and that was with us for 11 years. So this is a virus which has a different hemagglutinin, different from the H1, and this H2 virus with a different neuraminidase N2 was circulating for 11 years. And then in 1968, yet another pandemic strain emerged, and that actually, this H3N2 virus, is still with us. Now, in 1977, another H1 virus, or another H1N1 virus appeared, and this one actually is very similar to strains which were circulating in 1950, and this is really very surprising, and that virus was with us for about from 1977 to 2009, and this virus was replaced by what we refer to as a pandemic H1N1 in 2009. So at the present time, we have H3N2 viruses, we have H1N1, pandemic H1N1, and we have influenza B viruses. Influenza B viruses do not occur in terms of these different subtypes, which we have seen with respect to influenza A viruses, but B viruses also change, and that is shown in the changing color. If you look at different years, you have slightly different colors in terms of the influenza B virus, and what this should illustrate is that we can get reinfected in about if you get infected in 2016, for example, in 2 or 3 years, the virus has changed sufficiently that by 2019, probably we can get reinfected with an influenza virus because it has continuously changed, and that is referred to as antigenic drift. Whereas in terms of the influenza A viruses, when a new hemagglutinin such as H2 or a new hemagglutinin such as the H3 occurs, we are talking about a new pandemic strain, and that is referred to as antigenic shift. Now, we already talked about the 1918 situation, and there we had quite a lot of death, as I said, in terms of the overall, the estimates are up to 100 million. People died in 1918, 1919. In the US, these are illustrates the severity of influenza pandemics, and those are deaths in terms of the United States. As I said, we had about 750,000 in 1918. In 1957, this was a much milder one. This was the appearance of the H2 on the 10th, 70,000. The H3 was even milder in 1968, and the 2009 pandemic yet even milder than that. So this is quite important in terms of the different influenza viruses. Coming back to the 1918 one, I should have mentioned before, namely that the broken line shows that we don't have any viable viruses from the period before 1933, and the 1918 virus is actually an extinct virus. However, it was possible by a sequencing technology, and this was developed by Jeffrey Taubenberger, who was at the time at the Armed Forces Institute of Pathology in Washington, and he's now at the NIH. So by using material from soldiers who died in 1918, and the slide shows a lung block, the sort of translucent material here is actually a paraffin block, and the dark material, which we can see here, is material from the soldier who died in 1918. And by using this material and doing polymerase chain PCR reaction sequencing, it was possible by Jeffrey Taubenberger to actually get the sequence of this 1918 virus. And with the technology which we developed here at Mount Sinai, it was then possible And we refer to this technology as reverse genetics because we are making the RNA is the actual genome, which we talked about it. There are 8 RNA segments, which I discussed before. But then by using DNA and transfecting, putting into cells the 8 RNA segments in red here, which express the different RNA segments, and then also in terms of the protein expression plasmids in blue. Transfection with these 12 plasmids actually then resulted in the rescue of these viruses, and this was then a paper which was actually quite well received. The Lancet, the very important British medical journal, selected it as the paper of the year 2005. So more than 10 years ago, we were able to reconstruct the extinct 1918 Spanish influenza virus and characterize it in terms of its virulence and in terms of its different properties. So we actually know quite a lot about this virus now because we have it in the laboratory. We have to work with this virus under special conditions, which are referred to as biosafety level 3 conditions, where we can make sure that the virus does not escape and does not cause any harm, and this is done under very careful conditions. So this is the story about the 1918 Spanish influenza pandemic virus, which we reconstructed in the laboratory, and Terry Tumpy at the time he's still at the CDC, was able under very stringent biosafety conditions to get the first complete virus with all the 8 genes from the 1918 virus. Now, we have talked about the different influenza viruses which infect humans. Again, we have only H1, H2, and H3. But there are other viruses, particularly those which come from birds, avian influenza viruses, and they have been shown to actually occasionally to infect humans. And so we have, for example, if we look here, H5N6, in terms of the 2016 in China, this virus H5N6 was identified in China, and we have also, for example, an H5N1, which is shown here. This was a famous bird virus. We're going to talk a little bit more about this, but we have also a H7. We have 4 different H7s actually. We have the H7N9, we have the H7N3, we have the H7N2, and we have the H7N7. So one can see there are quite a lot of these avian influenza viruses, and they have been shown to infect actually occasionally humans. This is something which many people are worried about, particularly if we talk about the bird flu. This is the H5N1 virus, and this virus has actually caused a lot of different problems in the poultry factories, particularly in China. And we are actually very nervous that this would be something which we could jump into humans. Trying to move forward. So we have a slide here which shows us the distribution of these avian influenza viruses, and we can see that in yellow that these avian influenza viruses have been found in wild birds, in poultry, and as we can see in terms of the red squares, there were also H5N1 cases in humans. Quite a lot of people actually died as a result of these infections. The number of cases is probably much higher than is being shown here, because for the WHO only recognizes cases which are confirmed by laboratory diagnostics here, and they have to be shipped to either the US, to the Center for Disease Control, or to London. And one can certainly understand that there were many more cases of subclinical infections in these different countries, whether it was Vietnam or in China. So it is quite interesting that these H5N1 cases have been reported. They are real. It is likely, however, that these human cases are the result of large doses which are inhaled by people dealing with infected birds, infected chicken, and so far there is no evidence that the virus has been transmitted from one person to the other. So these are very potential new pandemic strains which might jump into humans. So what can we do against these different influenza viruses? And we have actually two different classes of FDA-approved influenza virus drugs. And these are the so-called ion channel blockers, amantadine or rimantadine. And these are only effective against the A-type viruses, not the B viruses, but there are also many resistant influenza virus strains, resistance against amantadine, so that the only real drugs which we have are neuraminidase inhibitors, and there are two types, zanamivir and oseltamivir, and they are effective against both influenza A as well as influenza B viruses. So this is something which is important, and we should be aware of these different drugs which we have. They are probably underutilized, and particularly in cases of severe influenza virus infections, we should really think about these antivirals, which are fairly safe, very safe, and represent a modality in terms of trying to treat and also prophylactically use them against influenza virus infections. The second major sort of tool we have against influenza virus infections are vaccines, and that's sort of what I'm going to highlight now, starting with the different types of influenza virus vaccines which we have. We have inactivated influenza virus vaccines, so they are not inactive, but these are viruses which are grown up and then inactivated by formaldehyde, so that the virus is not infectious anymore, but the protein is actually, when it is being administered by injection, it can actually induce an immune response which is protective. There is also FDA-approved an attenuated influenza virus vaccine, which is given through the nose intranasally, and that is a live virus, but it is highly attenuated, meaning very, very weak. It barely replicates, but it actually can induce a very good immune response in the nose and in the upper respiratory tract, which is important because that's the area, the location where the influenza virus is replicating. And then more recently, a third class of influenza virus vaccines has been FDA approved, and that is a recombinant protein, the hemagglutinin, and that protein is being made in baculovirus, by baculovirus in insect cells, and it is injected also, and this is a quite effective vaccine against the hemagglutinin and therefore against the virus. and particularly in terms of people who have allergies against egg products, because both of the inactivated and the attenuated strains are grown in embryonated eggs. The recombinant one has never seen an avian cell, and therefore people who are allergic to avian proteins can very safely use this recombinant influenza virus vaccine. Now, there are, as probably everyone knows, the vaccines for influenza virus, or against influenza virus, are good. However, they are underutilized and they are not perfect. They are not 100%. And that is one of the reasons why we are looking for a better vaccine. Specifically, you have to understand, I was mentioning before, we have 3 different influenza viruses circulating at the present time. We have H1N1 viruses, we have H3N2 viruses, and we have actually 2 different kinds of influenza B viruses circulating. If you look at the names, for example, if you look at B. Brisbane here, that means it's a virus, an influenza B virus, isolated first in Brisbane, isolate number 60, and was isolated in 2008. And as you can see, for the influenza virus vaccine formulation for 2016, 2017, the vaccine which we're going to get starting in the fall, has actually 4 different components. Unfortunately, again, an H1N1, an H3N2, and 2 different influenza B virus components. Now, unfortunately, this variability in the virus forces us to change the vaccine formulation very often. And this slide here shows, just look at the red and the black. This is so from 2000 to 2010. We have an H1, an H3, and a B virus component. And the different years show in red that the virus for the vaccine had to be changed from the previous year. So in many instances, 2 out of 3, or at least 1 out of 3, for example, in 2001, 2002, have had to be changed. There's only 1 single year, which was in 2003, 2004, where there was no change from the previous vaccine formulation. However, there is, for example, 1 year, 2008 to 2009, where all 3 components had to be changed. So this is something which is important and makes it very difficult to manufacture influenza virus vaccines, because one has to take into account the continuing change which we discussed before. It is also quite disturbing that in the case of the 2009, the red curve shows when the pandemic strain actually hit the United States. We had the majority of the influenza cases were already in October, November, and the blue line shows when the vaccine was actually shipped. And then it takes distribution, shipped from the manufacturers, and then it had to be distributed to pediatricians, here, to Walmarts, pharmacies, etc. And then it takes another 10, 15 days until the vaccine really works. So this is really something quite dramatic in terms of something as important as a pandemic strain. We are not able to really manufacture the vaccine strain for the 2009 pandemic in sufficiently fast so that it had an impact, a positive impact, or an impact in terms of avoiding influenza. Fortunately, this pandemic was very mild, and we actually were, so to speak, lucky. So now, in terms of the difficulties of the vaccines, one is they have to be changed constantly. In a new pandemic, we are not ready because it takes too long to manufacture it. And then also many times the vaccine strain doesn't match the circulating strain, because the decision of what kind of strain is actually added to the vaccine is made about a year before one actually vaccinates, and therefore many times the circulating strains are not what we would like to, are not matched very well to the vaccine strain. Then the vaccine efficacy is actually lower. So therefore the question is, how can we do better? And the answer to that is a universal influenza virus vaccine, one which would be effective against different strains and would be against different types and subtypes of influenza virus. So what are we doing in the laboratory? Sorry. And let me specifically say right away, the data I'm going to show are all in the laboratory, so we do not have a universal influenza virus vaccine at this point, but it should show us what we are thinking could be a solution. And the way we are doing this is actually a collaboration between 3 different laboratories at Mount Sinai. This is Florian Kramer's laboratory, Adolfo Garcia Soster's laboratory, and my own. And so the idea is that we are trying to make a vaccine against the conserved region of the hemagglutinin. So the hemagglutinin is an important protein on the outside of the virus, and we found, and I don't show all the data, but that there are 2 domains. So we have to think about the membrane of the virus is actually down here, and the hemagglutinin is sort of inserted into this membrane. There are about 1,000 of these spikes per virus particle, and the stalk, which is in green, is actually very much conserved, but it is also not very immunodominant in terms of immune response. In contrast, the head, which is shown here in brownish color, is actually more immunodominant. It is, we make more antibodies against it, and unfortunately it's also changing quite a lot. So this is what we are seeing in humans, and we hope that a vaccine which consists of a chimeric hemagglutinin, which has the same stalk as the one we have seen already, but a different hemagglutinin in terms of an H5, which we as humans have never seen. There is a hope that such a vaccine, which would induce an immune response against the stalk, and not much against the H5, because we have never seen this. So it's like being naive towards this head. And by then giving a second vaccination with yet another vaccine, which in this case has a different exotic head, like an H6, we would hope that there is some amplification of the B cells against the immunosubdominant stalk of the hemagglutinin. And we would hope that a lot of these antibodies against the stalk would then protect us in terms of all different strains belonging to a group 1 hemagglutinin. If you remember this one of our earlier slides, we would like to make also a vaccine against the group 2 hemagglutinin, as well as the influenza B virus stalk component. And so having those 3 components, a group 1 stalk, a group 2 stalk, and an influenza B stalk, we should be able to actually make a better vaccine. So this here again shows us that we have a situation here now where we are going with into animals. The animals are, and in this case we are talking about mice, and the mice have never seen influenza. They are not a reservoir for the infection, and of influenza, and we can see we have to prime the mice with the DNA, and all we do is we're using here again a irrelevant head of the hemagglutinin, but what is important is we are priming with a particular stalk, and then we are boosting with two different vaccines, again a different head with a chimeric H6 and a chimeric H5, and then we challenge with a virus which has either different from 1934, 1947, 2009, or even a completely different like an H5 or even an H6 hemagglutinin. So again, by vaccinating, in essence, with a vaccine that amplifies the conserved regions of the hemagglutinin, we hope that we will make we induce in humans a vaccine which is quite protective. And this is shown here, where we have again some antibodies against the stalk. If we come with the first vaccine, we get a different head response, but more in terms of the stalk, and yet more in terms of the stalk after the second vaccination with a chimeric H5. So this is important. We can show now that in terms of the vaccination with the chimeric constructs is actually leading to a protection. We are using here a 1934 stalk in the mouse, and we can protect against the virus, which is that's almost 70 years later. We get complete protection. We are challenge these mice, they don't lose any weight. The black curve shows what happens when we have no vaccination, and the control is in light pink. So this is sort of showing us that over a long period of time in mice we get complete protection. We can also show that we can protect, in this case, against H5 challenge or actually an H6 challenge. We can also protect these mice by a vaccine protocol or approach or strategy which I just discussed. So this is quite important. We were also able to show that these mice are the mice are protected against different subtypes. We can also show that we can do similar experiments with a group 2 stalk, and we were also able to show that the mechanism by which this happens is through antibodies by a passive transfer. Now, very specifically, we showed that these broadly protective stalk antibodies mediate their antiviral activity by a mechanism by which the antibody is actually let me show you this here. This is a model for optimal induction of antibody-dependent cytolytic, cytotoxic mediated or cell-mediated cytotoxicity, ADCC, antibody-dependent cell-mediated cytotoxicity. And these are effector cells. We have these antibodies here which go against the stalk. And we found actually very recently that by having the interaction between the FC portion of the hemagglutinin and the FC receptor on effector cells, and a second contact point, which is sialic acid of the effector cell and the head of the hemagglutinin, we get actually induced antibody, ADC, antibody-mediated cell-mediated cytotoxicity. And this is a very interesting mechanism. We have at this point, the NIH has made preparations of these new vaccines. So this is an H5, chimeric H5/N1. And we have also, these are about 10,000 doses each in terms of the chimeric H8 here. and we hope that these kinds of vaccines will be introduced into into humans, and that we can measure the immune response against such vaccines, and hopefully they are as protective as we were able to show in animals. We have only shown this in mice, we have shown it in guinea pigs, and we have shown it in ferrets, but obviously what is important is whether that kind of vaccine approach will also work in humans. So in summary, let me then sort of repeat or highlight what I have said. We are trying to make a universal influenza virus vaccine by reducing the immunodominance of the hemagglutinin head and thereby increasing the immunogenicity of the hemagglutinin stalk. And we have to remember that the hemagglutinin head is really what changes dramatically, but the stalk is much more conserved. And if we can sort of vaccinate against this conserved domain, we should be able to make a immune response which would protect us against many different challenge strains. And obviously these are difficult vaccines. They also have to be long-lasting, and also I haven't really discussed this. We also hope that by this redirecting of the immune response from the head towards the stalk, we should also make better immune responses against the neuraminidase, which is the second glycoprotein on the surface of the virus. And by having antibodies against the neuraminidase, which is also much more conserved than the head of the hemagglutinin, we hope to make a very efficient vaccine, which should protect us against all kinds of antigenic variants, including new pandemic strains, which there is no doubt that such viruses will occur in the future. So this is, again, this vision we have for a human universal influenza virus vaccine. Again, we have not been in humans. This is a very long process, but we hope that by redirecting again the immune response away from the head by using irrelevant heads, which we have never seen, we want to amplify the immune response against the stalk of the chimeric H5.1 and chimeric H6.1, and thereby protect against the different variants as well as different subtypes. And we believe that we will need a group 1 stalk, a group 2 stalk, and also an influenza B virus stalk. And again, this is work which we are doing in a collaboration, Florian Kramer's laboratory, Adolfo Garcia Soster's laboratory, and my own. Thank you very much, and I'm happy to answer any questions.
And thank you so very much, Doctor, for your informative presentation. Before we get started on the question and answer portion of the session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we're going to try to answer as many questions as we can. And our first question for you, Doctor, is this. Was the difference in numbers of death due from the H1N1 virus, others due to the vaccine that was not widely used aiding, or was it more due to the advances in healthcare in general, or was the risk hype and overstated by the media?
All of the above. It's probably all of the above. The vaccine, as I said, the current vaccine we have is not perfect. And the CDC actually said that the live virus vaccine this year was not as effective, or last year really, wasn't as effective as it should be. So there is that in terms of the live virus vaccine for last year, and the CDC is actually recommending the use of inactivated vaccines. We had in the years before mismatches in terms of the H3, so that was not optimal. So clearly this is not a perfect vaccine, but I would still strongly urge people to get vaccinated. It's the best thing we have. It's the only wheel in town. And again, we are working in the laboratory to make better vaccines, but as long as we don't have that, we have to accept what we have, and they are better, the vaccines, than some of the press releases make us believe.
Thank you very much, Doctor. In H5N1, for example, what do H and N signify?
The H and the N are the hemagglutinin and the neuraminidase. And those are the glycoproteins on the surface of the virus. H5N1 was only found in essence, in poultry. There were some occasional infections in humans, but this is not a virus which, the H5N1 is not a virus which is really easily, or is not really transmitted, is not transmitted from human to human. So this is I always say, if you are a chicken, the H5N1 virus is very dangerous, but in humans, so far, we have not seen transmission of H5N1, hemagglutinin subtype 5, neuraminidase subtype 1 in humans, from human to another human.
And thank you again. And our next question is, are there any serious adverse effects, such as autonomic dysfunction, that have been seen after the influenza vaccine?
The influenza virus vaccine is a very, very safe vaccine. When people get a flu shot and then they say 2 days later I got the flu, it is utterly impossible to get influenza from an inactivated influenza virus vaccine. There is no live virus. People can get 2 days later the flu, but then they were probably infected before they got the vaccine, and it certainly doesn't have anything to do with the vaccine. Again, in contrast to what many internet sites tell us, this influenza virus vaccine is extraordinarily safe, and we should not hesitate to take it.
Alrighty, thank you very much, Doctor. So we do have a couple more questions here, and if you would continue to submit those questions, you definitely would be able to do so. Can you comment on the mode of action of Tamiflu?
It's a very interesting question. Let me repeat it. What is the mechanism of action of Tamiflu? So this is a neuraminidase inhibitor. So it's an inhibitor of the enzyme, which is part of the virus, and it prevents the virus actually from leaving infected cells. So this is a drug, a very, very safe drug. It's a neuraminidase inhibitor. I briefly mentioned the two classes of antivirals. We have hemagglutins, and then we have the neuraminidase inhibitors. And Tamiflu is a neuraminidase inhibitor, which is very, very safe. It is given orally, and it prevents the spread of influenza viruses from one cell to the other. So when we give the drug, when a patient takes the drug, what happens is that the virus cannot replicate as effectively as it does it without the inhibitor, and it's a drug which prevents the effective spread from one infected cell to the other. That's a mechanism by which Tamiflu, a neuraminidase inhibitor, works.
Righty, thank you. And what is the efficacy percentage for 1 year range of the current flu casing in general population?
I'm not sure I understood the question. You are asking what the efficacy of vaccines is. Is that correct?
And yeah, that would be correct, doctor. Thank you.
So the question was, what is the efficacy of influenza virus vaccines? As we discussed today, there are several different influenza viruses circulating at the present time. There's H1, there's H3, and there's influenza B. And so the current vaccine also has to have components against H1, against H3, but also, and unfortunately it's getting more complicated, there are also two components against the influenza B virus. Now what happens is that some years we have more B than A. In some instances we have only almost exclusively H3. So the vaccine can have different efficacy against H1, against H3, and B. So there is no number which is correct for every year, particularly because the strains are different. And it also depends how you measure it. For example, many vaccines prevent death, but people still get sick. So if you are measuring the efficacy of the vaccine in terms of hospitalization and death, the vaccines are very, very good. But sometimes vaccines are measured by, is there any virus demonstrable in the patient, and then you get lower values. I would still say, in general, vaccines have an efficacy of higher than 50%. But there are some exceptions, and also it depends on the age of the cohorts, on the age of the vaccinated people. So there is no clear answer to your very good and excellent question. But think about it as something which is better than we don't have anything against HIV, for example. So clearly not taking the vaccine, particularly because it is so safe, is actually putting us at risk. So the vaccines are not perfect, but the presently available vaccines are very, very good. They are better than their reputation, so to speak. And again, the take-home message is that we are trying to make better vaccines in the lab. But so far, in the laboratory, for better vaccines in the future. But so far, we have not, there is no vaccine, a universal vaccine on the market, and it will take years until this becomes FDA approved.
All right, thank you very much, Doctor, and thank you everyone for your amazing questions. Doctor, do you have any final comments before we close?
To understand that it is important to understand that vaccines are really, really good. They are very, very safe, and we would really like to encourage people to think about taking vaccines, because even though they are not perfect and we are working on making them better, they are very, very good, and they are very, very safe.
And thank you very much, Doctor, for those closing remarks. And I also want to thank LabRoots as well for the continued sponsorship. Today's webcast will be available for on-demand viewing through December 7th. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.